Okay, there we are.
Great. So welcome to this webinar series on microglia. I'm Guy Brown and my co-host is Claire Butler. This is a series of monthly talks, about 45 minutes by European scientists talking about their research on microglia, followed by a Q&A session. And today's talk is by Professor Trevor Owens from the University of Southern Denmark, and he's going to be talking about microglia as modulators of CNS inflammation. So please, during the talk, if you think of a question, just write it in the chat function and send it to all while the speaker's speaking, and then we'll convey those to the speaker at the end of his talk. But during the talk, please keep your microphones muted and cameras off in order to preserve bandwidth. But I'm now going to be handing over to my co-host, Claire, who will introduce the speaker.
Great, thank you, Guy. Okay, so a little bit of an introduction on Trevor. So Trevor Owens is a professor of neurobiology in the Institute of Molecular Medicine at the University of Southern Denmark. He obtained his PhD from the University of Ottawa in Canada in 1981. Following postdoctoral training in London and Melbourne, he returned to McGill in 1987 and in 1990 joined the neuroimmunology unit of the Montreal Neurological Institute, where he became a professor in 2001. In 2004, he was appointed professor at the University of Northern Denmark, where he has been a leader of the neurobiology research department since 2010. His laboratory focuses on animal models of demyelinated diseases and specifically on interactions between immune and glia cells in the brain and spinal cord. He has published 124 papers in peer-reviewed journals. So with that, I'll let you go. Go ahead, Trevor.
Well, thank you very much, Claire, and thank you, Guy. It's marvelous what you've done here with this series. I hope I can match the previous talks. I'll just share my screen, and here we are. So I hope you can all see that. Tell me if there's a problem.
That's perfect, Trevor.
I think I have to move this away. So I'm going to talk about, it's a sort of a generic title, microglia as modulators of CNS inflammation. Microglia are tissue-resident macrophages, and like all tissue-resident macrophages, they derive from yolk sac precursors, different from many tissues. They derive exclusively from yolk sac precursors. Microglia are the tissue-resident macrophages of the brain. Therefore, all tissues have resident macrophages, and the brain is not different in that regard. So these are then yolk sac-derived CNS-resident macrophages. They're self-renewing, as we know from work from Bahara Ajami with Fabio Rossi, or from Julia Brutkar with Ari Weissman. And they renew from endogenous precursors without normally, except for experimental or traumatic conditions, they renew without participation from peripheral myeloid cells. And work from Masaki Ueno or Rosa Policelli, and then Dorothy Schaefer with Beth Stevens has shown us they play important roles in CNS development, contributing to neuronal survival and synaptic pruning. They're considered to be a homeostasis maintaining cell. They also contribute to protection by responding to pathogens and to injury. And they are implicated in neuroinflammatory and demyelinating diseases, and their roles vary depending on context. Now, that's a point that's going to be a recurring theme through the presentation I'm going to give. Now, why did that not work? There. So discriminating microglia from other CNS myeloid cells has been a constant for over a century, starting with the morphological description from Del Rio Ortega, and then introduction of immunohistochemical markers, surface markers. But right away it was clear that nothing that microglia expressed on the surface that could be recognized by an antibody was exclusively expressed by microglia. And even as we develop new markers, and there are claims for microglial specificity, there's always a paper that says that a macrophage can express the same. The image shown is from 1991, illustrating CD11B and CD68. The situation is complicated for those who want to study microglia, for instance, to isolate them or to do functional studies, because the brain, the CNS as a unit, contains many populations of myeloid cells. Microglia are the only parenchymal myeloid cell, but there are many other tissue macrophages, and including chorioplexus, perivascular macrophages, meningeal macrophages, and we need to be able to discriminate those. And the consensus mechanism for this, particularly for their isolation, is to use flow cytometry, and relying on the fact that microglia express a lower level of the hematopoietic marker CD45 than the other myeloid cells, which either infiltrate or are constitutively present. And this approach has been taken to in a feedback way, validate the approach by showing transcriptomic signatures, as Oleg Butovsky and colleagues have shown us, signatures for the homeostatic and activated microglia that actually do identify the population, but that analysis is done after they've been isolated, so it's a sort of a self-fulfilling prophecy in one way. But I'll be describing more about this. This is P2RY12, TMEM119, FCRLS. So it is possible to discriminate them, but it's always a caution to be careful that we're not confusing two populations. And this nice image from Amanda Sierra shows the multiple functions of microglia. We are most interested, and I will talk today mostly about their role in inflammation, their role as phagocytes, clearing debris, and their role in myelination and remyelination. Now, some microglia express a marker, an integrin complement receptor, CD11C. This is somewhat taken for granted now, but I want to go back through where that came from to lead up to where we're going forward, because my lab in Montreal were involved in showing this. We came around the same time. We published a year after Oleg Butovsky and Michal Schwartz identified CD11C microglia in Alzheimer's plaque-associated cells. We identified CD11C microglia, we being Alicia Babcock, who was looking in axonal lesion-induced microglial activation, and then Leah Remington identified similar cells in cuprosome demyelinated corpus callosum, and that's what we published. Couple of important points. One is in the unmanipulated tissue, one essentially doesn't see these cells. There's one. And I'll come back to that point. Two, we had in marker sharing CD11B and CD11C, which convinced us that they might not be dendritic cells, and they didn't particularly associate to laminin expressing vessels, so they didn't look like they were coming from blood. Flow cytometry then showed that these the black dots are the CD11C cells sitting in the cluster of CD45 dim microglia, numbers increasing with time of demyelination and falling away again when the demyelinating toxin is removed. Note that the black dots are sitting at the top end of this cluster, and this at the time allowed people to express skepticism that perhaps these were not microglia, perhaps they were a dendritic cell. This just shows the kinetics of their expression, as I have already run through, and the reduction when we return to homeostasis. Nicely for us, a few years later, Marta Ola and Eric Bodeker's lab also showed CD11C positive microglia in cuprous demyelinated corpus callosum, and implicated them in myelination, which we afterwards have done as well. We have also seen CD11C microglia here co-stained with IBA1 in EAE, and CD11C positive microglia in mouse models of neuromyelitis optica. And these are the two that I'm going to talk about today, EAE and NMOSD. I'll come back to details on those. So we then move from Montreal to Odense and take on two PhD students, Agnieszka Wlodarczyk and Morten Loebner, and they studied the antigen presenting and related functions of microglia and infiltrating CD11C positive cells to compare them, addressing really the question of how different are they from each other and how distinct are the microglia. So going back to the original paper, CD11C positive microglia, Leah with Simone Zentner and Alicia showed that they're very good antigen presenting cells for inducing a secondary T cell proliferative response. So we said, aha, these are antigen presenting cells. Therefore, they're probably implicated in inducing T cell inflammation in the CNS. First, we needed to show that they were quite distinct from the infiltrating CD45 high cells. Here are the CD45 low, CD11C positive cells, microglia, and flow cytometry identified that even though that, yes, they do have a higher CD45 than the CD11C negative microglia, these are clustering together in the CD45 low gate, and their numbers increase in EAE. They express CX3CR1 and don't express CCR2, supporting a microglial specific status. And then our first surprise, they don't express, and this is in EAE, the cytokines that you would expect an antigen-presenting cell to express if it was activating a pro-inflammatory T cell, Th1, Th17, whereas the CD45 high cells, which in this paper we termed CNSDC, do. So they don't, even though they induce proliferation, they don't look like very useful antigen-presenting cells for inducing a T cell inflammation. We asked, did they express type 1 interferon, wondering if they were not inflammatory for T cells, perhaps they were anti-inflammatory, and interferon beta is a recognized therapy for MS, and we wondered about that. But it turns out that both the CD11C positive and CD11C negative microglia express interferon beta, and they respond. This is IRF7 and IRF3. And so they didn't look particularly different in this regard either way. We, using old-fashioned and out-of-date M1, M2 thinking, we looked at some function-associated markers, and we could discriminate the CD11C negatives from the CD11C positives. CD11C positives expressing ARG1, INOS, and interestingly, IGF-1, insulin-like growth factor 1, growth factor implicated, for instance, in myelination. They're amongst the myeloid cells in the CNS, the CD11C positive microglia, and this again is EAE, these are infiltrating CD45 high cells. Amongst the myeloid cells, these are not just a potent source of IGF-1, they're really the only source of IGF-1. We know there are other cells in the CNS making IGF-1, but amongst the myeloid cells, the CD11C positive microglia are where it comes from. So these are microglia that show regulatory characteristics, and so then we asked, could we induce the regulatory microglia for therapeutic benefit? And now Agnieszka is joined by, or was joined by, is still joined by, Anouk Ben-Mamar-Badel, a PhD student who's just about to defend her thesis, to look at whether we could induce these microglia and neuroprotection by stimulating the CSF1 receptor, colony stimulating factor 1, macrophage stimulating colony stimulating factor. This receptor binds two ligands, IL-34 and the nominal CSF1, and induces a variety of functions, migration of cells, myelin repair has been implicated by Nathalie Laflamme with Serge Rivet, and maintenance, development, proliferation, and maintenance of microglia. And maintenance actually dramatically is shown by the fact that if you block signaling through this receptor, you eliminate microglia. And I'll come back to that. We've used this in a subsequent set of experiments. But for now, I'm going to describe what happens when you apply these CSF1 receptor ligands to a mouse with EAE. And applying the ligands was done by intrathecal injection. This is a picture of a brain isolated from a mouse. This would be the nose of the mouse, and this is the spinal cord beginning here. And here at the base of the cerebellum, where the cerebellum sits over the brainstem, is the cisterna magna, cerebrospinal fluid filled chamber compartment that you can access. We bend the needle to be able to do it in an anesthetized mouse. And what Agnieszka and Anouk did was gave 3 daily injections of recombinant IL-34 or recombinant CSF1, and after that, on the 4th day, counted CD11C positive microglia, and sure enough, their numbers doubled, even 2.5, tripled nearly. Statistically significant here, almost significant here. So they can achieve an increase in the number of these microglia. What was the functional outcome? The functional outcome was inhibition or suppression of EAE. Now, the way this experiment was done was to immunize mice for EAE, and the inclusion criterion for a mouse, the entry criterion for the mouse to enter the experiment, was that it show clinical symptoms of EAE, approximately grade 2, corresponding to a floppy tail, and that's defined as day 0, from which mice increase their clinical severity to grades 3, 4, and 5, depending on the experiment. The mice that received recombinant IL-34 or CSF1 did not show this progression. There was a very small progression here, an increase in severity, but then there was a statistically significant loss of increase of severity, also reflected in the maximum clinical score, which was statistically significantly lower for both IL-34 and CSF1 treated animals. This was also shown as reduced demyelination when the animals were examined at 4 days. This is MOG staining. These are areas of loss of MOG staining outlined in, with a line to help the reviewer. You do not see these areas of loss of MOG staining in the IL-34 or CSF1 treated mice. That's quantitated here as a demyelination score. Again, significantly reduced demyelination in the CD11C positive microglia induced mice. Correspondingly, the loss of oligodendrocytes that one sees in the control animals that have progressed EAE, there is a significant loss of no-go-A positive oligodendrocytes. That is not the case when the animals have received IL-34 or CSF1. So what is the mechanism? We don't know. We know that these cells make IGF-1. I've showed you that. IGF-1 would be a logical candidate for mediating this protective effect. We would like to think that's the case, but unfortunately I don't have any data to show you on that for the moment. We are working on it. So having shown that, we then asked a couple of questions. Do these microglia play a role in CNS development, since microglia are known to play a role in CNS development? And can this be exploited for therapeutic benefit? And, you know, I wouldn't be asking the question if the answers weren't yes. This is building on an observation that Agnieszka made, is that these cells can be found in the developing brain. And she did this work then in collaboration with colleagues in Groningen and in Mainz, as well as others. Her observation was that, as said, the CD11C positive microglia emerged during postnatal neurodevelopment. And you can see that here as quantitation of the proportion of these amongst the microglia, or their absolute number. And in both cases, starting from birth, there's an increase in their number to rather high percentages. And a dramatic fall in these percentages and numbers as the animals age to become adults. And remember, I showed you in the original description of these in the cuprizone system that in the unmanipulated corpus callosum you couldn't find these cells. The same is true when you do a isolation of CD45 low cells and count the CD11C positives, as are here, which are also CCR2 negative, repeating an observation from before. Whereas the CD11C negative microglia basically plateau and continue at a similar number through the aging of the mouse. And they co-express CX3CR1 as shown before. We find them in the corpus callosum and the cerebellum, regions of white matter, myelination, co-staining with CX3CR1, as I said, co-staining with IBA1, and not particularly associating with laminin, so not looking like they've recently come from a blood vessel. And then transcriptomic analysis, RNA-seq analysis of isolated cells, isolated now as CD45 low, CD11C negative versus positive cells, and then bulk RNA-seq. The immediate observation is that the neonatal microglia, that's here the CD11C negative and here the CD11C positive, they have upregulated many genes that are not seen upregulated in microglia isolated from an adult, a normal unmanipulated adult brain. Enormous gene upregulation in both populations, but particularly in the CD11C positives. And these include many growth factors and signals for guidance, myelination, astrocyte differentiation. GP-NMB is the top one, osteopontin's the next, IGF-1 is here. Double cortin is somewhere down here. So these are genes that look like they're associated with differentiation and tissue remodeling. So then we asked, IGF-1, I told you that they express that. They're a major source of IGF-1 in the neonatal CNS, as they are, as I showed you earlier, in EAE. And Agnieszka used a Krelox approach to selectively ablate IGF-1 in CD11C expressing cells, most of which are microglia in this context. And this significantly reduced the thickness of myelin 3 to 4 weeks later, as can be seen by the altered G ratios. So yes, these cells do play a role in primary myelination through their production of IGF-1. Then we asked, do CD11C microglia play a regulatory role in antibody-mediated disease? And we asked this because antibody-mediated disease has become discriminated from MS. We are looking at a mouse model for neuromyelitis optica, which used to be considered a subpathology of MS and is now recognized as a separate disease. And we've noticed some interesting aspects to this, which were followed up now by Agnieszka in collaboration with Reza Kharoushi, a research associate in the group, and with a number of others, including some old friends from Groningen. We took advantage of a system that was developed in the lab by Nazrin Asghari. Nazrin is a neurologist who did her PhD in the lab, and she is now a clinical professor at Sleosa Hospital in Zealand. And she innovated a system where She isolated antibodies from her NMO patients, purified IgG. Most of this is specific in most patients for a water channel aquaporin 4 that is expressed by astrocytes. And transferring this purified IgG with a source of complement into the CSF by the intrathecal injection here in the back of the head, under the cerebellum, into the CSF of the mouse. This results in NMO-like pathology that is strikingly similar to what you see in clinical NMO. Here showing demyelination after 7 days. We also assay astrocyte pathology, here shown by loss of aquaporin 4 staining. This is the intrastriatal injection. The black line marks the needle track, and the other line is outlining an area of loss of aquaporin 4 staining, which reflects loss of astrocytes. You can also stain for GFAP and see exactly similar pathology. So this pathology can be induced by a local stereotactic injection of NMOIG and complement. And the point of this particular study was that if the mouse into which the NMOIG and complement were injected did not express the receptor for type 1 interferons, the IFNAR knockout mouse, there was significantly less pathology. And that was a very important observation for us, because one of the things that distinguishes clinical NMO from MS, and which was part of the segregation of these as different diseases and not separate pathologies of the same disease, is that interferon beta, which is an effective therapy against MS, has no effect in NMO clinically. And here we show a requirement for type 1 interferon for the NMO-like pathology. So moving on from that, Agnieszka then did RNA-seq analysis on bulk RNA from the NMO tissue here, showing significant gene upregulation compared to RNA from brains of mice that received normal IgG and complement, and also downregulations compared to the normal IgG and complement. These mice do not show pathology. And here are some selected genes upregulated, MX1, CLEX7A. 75% of the genes that were upregulated in NMO were regulated by interferon 1, by type 1 interferon, as screened via the interferome database. So this is a very strong interferon footprint imposed on the CNS of the NMO pathology animals. Similarly, if we co-injected interferon beta with the NMOIG and complement, we got a significant increase in pathology. Now, don't be fooled here. There's a trompe l'oeil that easily fools you. This is not pathology. These are ventricles. The pathology is here. Needle track, it's a bit small, and it's significantly increased in the animals that received interferon beta. Whether you look at aquaporin 4 loss or GFAP, you can see a significant increase in loss of staining, which you do not see in animals that received normal IgG. The interferon beta exacerbates NMO pathology. It's got nothing to do with, for instance, complement, although complement is required. This is an interferon beta effect on NMO pathology. So then Agnieszka asked, what about the microglia? There is microglial, I think I've left something out. No. As I had said before, there is microglial activation in this NMO model, and there is, as expected, induction of CD11C positive microglia. And here you can see the quantitation of those as a proportion of the total microglia. This occurs in animals that received NMOIG and complement, but not animals that received normal IgG or nor in an IFNAR knockout animal. In other words, only if there's pathology do you see the CD11C positive microglia, and you see twice as many of them in animals that received interferon beta. RNA-seq identifies, principal component analysis identifies that these are distinct populations, the CD11C negative, the CD11C positive, compared to controlled animals. Nevertheless, they share the CD11C positive microglia share core signatures with the other CD11C positive microglia that I've described. And although they're distinct, they have the commonality of having 43, 50, approximately half of the genes that were upregulated in either subset were type 1 interferon regulated. And here comparing the genes upregulated in CD11C positive versus CD11C negative, and comparing those to genes upregulated in NMOIG compared to IFNAR knockout versus normal IG, there's significant overlap, and the pink line identifies a cluster of 45 genes that are shared between both microglial subsets and upregulated in NMO. PCR here confirming or validating a couple of them, IRF7, CXCL10. So there's a strong interferon footprint in the microglia as well as in the whole tissue, and that led to another question, which is what are the microglia doing? And this is the CSF1 receptor inhibitor, Plexicon 3397. Agnieszka has fed this to the mice, and this has removed the microglia. Here, the IBA1 positive microglia associated with the NMO lesion. You do not see them in CSF1 receptor blocked depleted animals. And importantly, you also do not see NMO pathology, seen either as loss of aquaporin 4 staining or loss of GFAP staining. Both are significantly reduced in the absence of microglia. The NMO pathology, the red bar, is the microglia depleted with NMO. The NMO pathology is dependent on microglia, as is the interferon response. Here are a number of genes, IRF7, CXCL10, MX1. These gene activations were suppressed in the absence of microglia. And then a curve that the system threw at us, which we didn't expect, interferon production itself, this is interferon beta, was not affected. Now, I spent some time telling you earlier that microglia, whichever subset, make interferon in an EAE context. Here, removal of the microglia has apparently had no effect on levels of interferon message. So we do not believe this means that these microglia were not making interferon, but it does mean that some other cell is making interferon. And that's one of those other things we'd love to be able to tell you more about, but we don't have enough data to say anything that we feel confident of at the moment. So the microglia are responding to interferon. They're necessary for pathology, and the interferon is coming possibly from microglia, but also from another cell in the system. So what we've shown then is that in contrast to EAE, microglia, and we have not discriminated CD11C positive and negative here other than to say that they're both there. In every other respect, they seem rather equivalent. So we wonder whether the CD11C positive microglia also are pro-pathogenic in NMOSD, distinct from EAE, whereby interferon acting on microglia results in signals that promote NMO pathology. This does not happen in the absence of microglia. And if it's the case that CD11C positive microglia are now behaving in a pro-pathogenic manner, is this a context-dependent function? So we have to engage now in a little bit of analysis. One thing I wanted to do here was to introduce a sort of a finger wag that's pointing at me, because we make the same mistake that many people make of guilt by association. I like talking about this one with Burkhard Becker. He also shares the same concern that finding activated microglia at sites of neurodegeneration and demyelination has frequently been used as a way of suggesting that they might be pro-pathogenic. Similarly, finding them in situations of suppression, you can draw conclusions, but the analogy that shows up at my lab meeting every week is the people most commonly found at a fire are firefighters. What are they doing? Well, they're putting the fire out, but there's more of them than anybody else. So function needs to be interrogated directly. Even though we do the best we can, and we did this for the CD11C microglia in the neonatal mouse, neonatal brain, and we tried with the plexicon, although we didn't discriminate CD11C and CD11C positive. We've had a go. We do, where possible, we interrogate function directly, but we, as many others, sometimes fail to demonstrate the effect of the presence or absence of any cell type. So following along on that principle, and wondering why I'm not advancing, there we go. We've compared CD11C microglia that we find in different pathogenic contexts. Specifically in this group of databases, comparing CD11C microglia with similar morphology, location. They tend to locate at the lesion, but if the lesion is MS or glioma or injury, the lesions can be in various places. If the lesion is an Alzheimer's plaque-associated lesion, they'll be associated with the plaques. The timing of induction depends on the model. There are shared markers, and one of them being CD11C, because I've defined that we're looking at CD11C microglia, CLEC7A, ApoE, IGF1, CSF1 comes up. The databases we have compared are 3 from Alzheimer's disease or Alzheimer's disease models and our own EAE RNA-seq data. They're variable. They come from different groups. They're publicly available databases, and Anouk has put these together as a comparison to compare. These are APP-PS1 mice. These are 5X FAD mice, and as I say, these are our EAE mice. In some cases, the microglia were specifically identified as CD11C positive by staining. In other cases, we know that they express CD11C from the gene expression profiles. The methodologies for isolating cells varied. The methodologies for assessing the transcriptome varied, sometimes microarray, sometimes RNA-seq, sometimes single-cell RNA-seq. Nevertheless, a core of shared genes, of 89 shared genes, could be identified, shared by at least 3 of the subsets. And this overlaps, as I will point out in a minute, with other CD11C positives that I've talked about. Not quite the similar signature, but many of these genes are also found upregulated in microglia, in ALS, glioma, stroke, ischemia, brain injury. These are all contexts in which there is cell death. In purely inflammatory models, where microglia are activated by LPS or poly-IC, the literature that we have screened and our own data suggests that we do not see the same gene upregulation. So there's something about the CD11C associated gene signature that relates to, amongst other things, cell death. It's a speculation, but I'll show some of the evidence that supports it. We've also compared CD11C subsets from postnatal CNS. We've identified 4 studies that have looked at microglia in the between P0 and P10 postnatal brain. The microglia have similar amoeboid morphology, location, corpus callosum, cerebellum, and again we see familiar faces, familiar names: CD11C, osteopontin, IGF1, CLEC7A. Sometimes the cells are identified by MAC3. Again, variability in isolation methodology, identification of the cells with different markers, the transcriptome analysis, single cell protocols versus bulk RNA-seq. This is actually microarray. Again, we see a shared cluster. This is 39 genes that are shared between at least 3 of the populations, and there's a core of 11 genes expressed by all of them. Here are the genes that are shared now by the PAMs, ATMs, found in the microglia, and our own CD11C microglia. This is the core of 11 genes. ITGAX is CD11C. Here's IGF-1, CSF-1. 28 are shared by at least 3 of the subsets, and you can see CLEX7A and others that I have mentioned, LGALS1. The ones that are in bold are also shared by the DAMs, neurodegeneration-associated microglia, and the CD11C plaque-associated microglia from the previous slide. So there's a lot of fingerprint sharing between CD11C microglia, not always called CD11C microglia, called DAMs, called ATMs, called PAMs. So in this paper that Anouk and Agnieszka put together, it was we proposed that based on this shared core transcriptomics signature, we proposed that all these populations with different names studied by different groups in overlapping contexts, they represent a common subset to be detected under variable experimental conditions. We call them CD11C microglia because that's how we found them. That's how we identify them. They all express CD11C. We could probably come up with other names, but whatever the name, there's a common subset. The evidence suggests a functionally common subset of microglia. So we see that what's happening, beginning to summarize now, in the neonatal context, the proportion expressing CD11C is identified here in this pie chart. It can be as high as 20-25%. These are PAMs, ATMs found in the microglia, CD11C microglia. As the animal ages, there's a loss of proportion of these cells. We go to a homeostatic state. They then re-emerge in disease or aging. The inducing stimuli include, we think cell death plays a role. Oleg Butovsky in the original paper identified IL-4. In the later paper, in the APP-PS1 mouse, the TREM2 ApoE signaling pathway was implicated. We have, and I didn't go into this other than to summarize it now, we have data that the chemokine CCL2 plays a role, which is interesting because it's not acting through CCR2 because the cells don't express it, it's acting through another receptor. I've shown you the data that CSF1 receptor ligands play a role in inducing these cells, and there's also data published that SIRP-alpha CD47 signaling suppresses induction of these CD11C microglia, which all the way through express their core signature and are then re-emerge in the context of degeneration and aging, where in fact percentages can become quite high. What did I tell you then? I've told you that CD11C microglia have a regulatory glial antigen-presenting cell phenotype that can be re-induced or recalled by CSF1 receptor ligands, and that results in suppression of EAE with reduced demyelination, a suspicion that there may be remyelination, and I wish I had the data, but we don't, whether IGF-1 plays a role here. These cells are strongly represented in the neonatal CNS, where they're a major source of IGF-1 and critical for primary myelination. They can be transferred to mice with EAE, which results in suppression of disease, again reduced demyelination, again a suggestion for remyelination, and again a question mark whether IGF-1 plays a role. And then the CD11C microglia are upregulated in experimental NMO. They're interferon responsive, and interferon is necessary for the pro-pathogenic role they appear to play. They're necessary for astrocytopathology. And that's opposite to what we see in EAE. And so the question is, why the opposite roles in NMO versus EAE? So now a bit of philosophizing. This is a modification of a slide from a nice review by Vasilis Stratoulias and Bertrand Joseph with Marie-Ève Tremblay. It's a panel suggesting that a single precursor population can give rise to different subtypes, functional subtypes of microglia, depending on the inducing stimulus. So that, for instance, and I'm using it to suggest that CD11C microglia might become one thing in this disease, let's say EAE, and become another thing in that disease, let's say NMO, have a different presentation in aged animals, newborns, and a context-driven alteration of functional outcome. But there is another possibility, which is where I'm going to end, which I think is important to keep in mind. Another of the functional outcomes that our transcriptomic analysis has pointed to and was there from the very beginning is the role of microglia as phagocytes. And a phagocytic microglia can contribute to remyelination by clearing myelin debris. The same phagocytic function may not be so useful in NMO. So what we may be looking at is the same functional output, but with a different outcome, because phagocytosis of a damaged cell in NMO will generate the lesion that we measure, whereas in EAE it will contribute to remyelination, which we think is going on. So what I'm going to leave you with is the concept that we may be looking at the same cell wearing different hats. I'd like to thank everybody who did the work. In particular, Agnieszka, who is now an assistant professor in the group, Anouk, who I said is about to defend her PhD, Reza, who is a research associate professor, Joanna, who is unfortunately about to move on to something else, members past, and our other members present, Dina, who takes care of the animal ends of things, and Pia, who helps us in the lab, Nazrin, who innovated the NMO model, our colleagues at Odense University Hospital, our colleagues in Groningen, Leipzig, and Mainz, and of course, our funding, our source of funding, for which we are extremely grateful. So thanks to them, and thank you for your attention.